All right, let's go ahead and get started. Before we get started with the content, we have one announcement from our graduate TA, Tim.
So y'all have an exam on Thursday. We're gonna hold a review session tomorrow in this room, 1041 at 5:30 p.m. till 7. That's it.
Thank you. All right, so up to this point, we have talked about, again, taking a quick rundown of everything we've discussed about up to this point. We began talking about proteins and amino acids. We talked about different classes or families of proteins. We then jumped into immunology, talking about both arms of the immune system, the adaptive and the innate, different proteins that are involved with that, as well as different cell populations. We're going to be talking about a fairly central kind of giving away the title here, but a fundamental concept in molecular biology called the central dogma. And we'll be applying that or talking about it not with respect to a disease, but rather a process that's fundamental to immunology called antibody diversity. So there's been this recurring idea, especially when we've talked about adaptive immunity, with the ability of our antigen receptors, particularly our antibodies, to be able to recognize and identify millions millions of different kinds of antigens, having this library of T cells and B cells equipped to respond to an antigen that you have never seen before. And each clone of T cell and B cell that emerges during development is going to have its own antigen receptor that is poised to respond to any given antigen that you might encounter. And so when we think about the space in our DNA, the space in the human genome, to have millions of possible antigen receptors, the question might emerge: do we have different genes for each of these possible antigen receptors? The answer would be no, because a helpful number to have in mind is that we only have about 20,000 genes within our DNA. And so there's no way that among 20,000 genes that we could encode for every possible permutation of antigen receptors. And so there are mechanisms in place, excuse me, mechanisms that enable us to leverage the fact that we only have about 20,000 genes, but create millions of possible different antigen receptors. So we'll be talking about antibody diversity or antigen receptor diversity. Again, that'll be our application point of this concept of the central dogma. This is this overarching concept of how the different molecules in molecular biology relate to one another. And so the main actors here are going to be DNA, RNA, and protein. So DNA is transcribed into RNA. That process is transcription. You may have heard it referenced a couple times already. We're going to have an opportunity to talk about it in more detail in the next unit. But DNA is transcribed into RNA. From there, RNA, there's a particular flavor of RNA called messenger RNA or mRNA, where mRNA is translated into protein. And that process is called translation. And so there is another add-on in some ways where, while DNA begets RNA and RNA begets protein, we can also have DNA replicating to form more protein during DNA replication. When we talked back during the cytoskeleton lecture during mitosis, where there's this chromosomal segregation. During mitosis, during cell doubling, the DNA will be replicated in each subsequent cell that emerges. But the big picture here is that we have DNA that's transcribed into RNA and RNA that's translated into protein. When we talk about nucleic acids, the two flavors are going to be DNA and RNA. Both of them are going to be, whether it's during transcription or during DNA replication, both of them will be produced by using some DNA strain as the template, the strain that's being read to produce either more DNA during replication or to produce mRNA or produce RNA during transcription. The enzymes that allow this are called polymerases. And the nice thing about this is that there are the naming convention is pretty straightforward. DNA polymerases are what are used to produce DNA, and RNA polymerases are what's used to read the template DNA and produce RNA. And so we're going to be going over this in greater detail in the next unit, but we're just going to have an opportunity to just quickly, at a fairly high level, just glide over and take a quick look at replication and transcription. So when DNA replication occurs, again, DNA replication is producing DNA when reading DNA. So we have a double-stranded DNA here that is separated or melted. We have our template strand of DNA. What ends up happening is that because of how DNA polymerases work, there's a need for some sort of primer to begin this process, and that's usually an RNA primer. So we have DNA polymerase that comes in and produces a DNA strand that is complementary to the template strand. And then there's another flavor of a DNA polymerase that replaces that RNA primer with DNA. So big picture here is that there are DNA polymerases that read DNA and produce DNA. Okay, it's reading the DNA to produce something that's complementary to it. You guys may remember from previous biology classes that you've taken that there is base pairing between the nucleotides in DNA. We have A, T, C, and G. And so the base pairing of A to T and C to G is how there's complementary DNA strands. We'll talk about in greater detail about why those nucleotides specifically pair together, but it's just good for you to know that when DNA is being synthesized here, during replication, that it's reading a strand of DNA and producing a complementary strand to it. During transcription, instead of melting and separating the double-stranded DNA, what ends up happening is RNA polymerase kind of just comes in and wedges itself inside the DNA here. And what ends up happening is almost kind of like a zipper reading through double-stranded DNA. It's going to read a template strand and produce mRNA or produce RNA as it's reading the template strand. And so both of these polymerases are, what's common here is that both polymerases are reading a strand of DNA. It's double-stranded. One of the strands are going to be read. But what's different between these polymerases is that one is producing RNA, it's the RNA polymerase, and the DNA polymerase is producing DNA. Yes. We're not going to we're going to jump it. We're going to cover that in greater detail in about 2 weeks. So there's usually one strand that's going to be, this is not happening. So if you imagine our DNA is organized into chromosomes, that's how our DNA is packed in together, and so it can fit inside the nucleus. Certain regions of the DNA, this process of replication or transcription is happening all over a single chromosome. So one part might encode the template strand is going to be like one particular part where it's being read. And on the other side, there might be transcription going on in the opposite direction because there might be an adjacent gene nearby. And so like this is happening all over your DNA at a given moment. But how we distinguish the template strand versus the not template strand, we're going to cover that in a couple of weeks. I appreciate that question. Hold on to it. Keep that in your mind during spring break, and we'll come back to it when you get back. And so both transcription and replication use polymerases that read one of these strands of DNA, but DNA polymerase produces DNA during replication, and the RNA polymerase produces RNA during transcription. So we have referenced genes multiple times in this class, but it's going to be really important for us to help define what is a gene, what precisely molecularly is that when we talk about it. And so a gene is the entire nucleic acid sequence, DNA in this case, that's necessary for the synthesis of a functional polypeptide, functional protein. So if we take those 2 bullet points of central dogma, DNA transcribes into RNA, RNA is translated into protein. A gene is where some segment of DNA leads to the production of RNA that leads to some protein. What that means is that there's going to be some parts of our DNA that do not lead to that do not encode for some protein, and we're going to have a name for that in a moment. But this is going to be our working definition of how we define a gene. It leads to some functional protein or polypeptide. There are regions of our DNA that are transcribed, but do not encode for some protein. We would refer to that as non-coding RNA. The RNA that's produced there that doesn't encode for some protein, we would call non-coding RNA. And so those are, while they don't technically lead to the production of some functional protein, we do refer to those the sequences of DNA that encode for this non-coding RNA. We still refer to those as genes because these non-coding RNAs provide a really important function in genetics and in molecular biology that we're going to talk about in a subsequent lecture. And so particularly in eukaryotes, in mammalian DNA, our genes are our genes, these sequences of DNA that encode for some protein, exist within this huge sea or this huge expanse of non-coding DNA. Most of our DNA is non-coding. They do not encode for some protein. And so the 3 billion base pairs of DNA that we have, a very small fraction of it encodes for actual genes, encodes for actual proteins. And so there's going to be 3 key terms that we're going to take a moment to define when it comes to how we talk about genes. There are parts of our DNA that we refer to as introns, introns, exons, and extragenic DNA. So exons are the part of a gene. This is within a gene again. Exons are the part of our gene that actually code for part of the protein that's actually expressed. Okay. That constitutes about 1% of our entire DNA. Introns are the regions of DNA that lie in between the coding parts, the lie in between the exons. So introns lie in between exons, and I'll say that differently. Introns are the regions of DNA that do not encode for what is going to become the protein. So as an example here, in gene 1, we have 2 exons, A and B. In between exons A and B in gene 1, there's this black line in between them. That would be an intron. It does not encode for what's going to become part of the actual protein. Same is true for gene 2. We have 2 exons, A and B. They both encode for a part of what's going to become a part of the protein. In between them, exons A and B, is what we would refer to as the intron. Now, again, most of our DNA is going to be what lies in between genes, what we call extragenic DNA. So between genes 1 and 2, there's this segment here. This does not encode for any gene. This was what we'd refer to this as extragenic DNA. And also to the right of gene 2 and to the left of gene 1 does not encode for anything. We would also refer to that as extragenic DNA. This is about 3/4 of our DNA. What's noteworthy is that there's a lot of work being done now to identify important regulatory roles that some extragenic DNA provides, where they don't encode for anything, but they can actually help regulate and provide some feedback on how genes are expressed. But we're going to talk about that a little bit more in the next unit. Yes.
But you have is all of our extragenic DNA doesn't this really matter what it is? So are there conditions where you have an extragenic DNA mutation that
Yeah, that's a great question. So mutations, so there's both this there's this evolutionary protection where if there's some mutation that occurs within our extragenic DNA, that's not in some cases will not have any effect on our coding genes. However, there again is some extragenic DNA. So let me take a step back. So I had described earlier how our DNA exists in this form of chromosomes, how they're kind of packed tightly. We're going to talk about this in greater detail in the next unit. But we talk about how proteins fold and how it exists in 3D structure. DNA exists in 3D structure as well. And so this extragenic DNA can actually, when things are folded on top of each other, can actually affect how much transcription occurs, you can to help with some vocabulary. When you hear a gene being expressed, think of that gene being transcribed. When transcription occurs, that gene is being expressed. The expression of these genes can sometimes be regulated by extragenic DNA that can like fold back on top of them and can impact transcription occurring there. So does that help? So like a mutation in the extragenic DNA can actually feed back there. And so when we talk about exons, introns, and extragenic DNA, it's helpful to have a grasp of these concepts so we can go into greater detail about some concepts. I have two questions. I'm going to go three. I'm going to go here, here, and then there. Go ahead. Yeah. Yeah, so that's what I tried to provide. So I tried to provide like a general rule of thumb. So we define a gene as something that encodes for some protein, some polypeptide. An exception to that is that there are segments of our DNA that are transcribed, produce RNA, but the RNA does not get translated into protein. That RNA, we previously thought for a long time that we call it non-coding RNA because we thought it served no function. We've actually come to understand it provides a super important function to a degree that it was awarded the Nobel Prize about 2 or 3 years ago, the discovery of like what non-coding RNA actually does and the different flavors they come in. So now because of the important function that we understand non-coding RNA provides, we now will provide an exception where the DNA that encodes for non-coding RNA, we also consider a gene. So that's just an exception to the general rule. Does that help clarify? You want to go ahead?
What about like unexpressed? Like genes that are turned on and off, where do they fall in the exon, intron, extragenic category?
Well, because genes that are not expressed, they're still genes. They have exons and introns within them. They just don't get transcribed. So there's regulation where the DNA that you have is the same DNA in every single cell. However, some of that DNA some of those genes are silenced and not expressed. For example, you wouldn't want your skin cells to start producing bone, or at least the proteins that contribute to bone. So those proteins are silenced, but they're still there. It's just that they're regulated differently based on what kind of cell this is. They're still exons, they're still introns. It's just how those genes are expressed is regulated differently because of what that cell is. You want to go ahead? Is there a mutation that causes extragenic DNA to become a gene? I like that question. I don't actually know. Most of what we have come to understand in the last 10 years about like new understandings about extragenic DNA is and this goes to a question that was asked earlier about how they can contribute to the regulation of expression of actual genes, if they can enhance expression or suppress expression. But I don't know of any mutations that lead extragenic DNA to become an actual gene.
It's still those same 4 bases.
Right, and there's yeah, actually, now that I'm thinking about this out loud, sorry, I'm an external processor. There are lots of parts that make up a gene more than just introns and exons. Like there's things that mark like where the gene begins, where the gene ends. There's like lots of other nucleotides that go into what forms a gene. So there's like lots of other things that go into it that would need to happen that likely, if you were to calculate the probability of it, probably wouldn't happen.
It's just not just the.
Exactly, yeah. Go ahead.
Genes are only composed of introns.
Genes, so great question. Genes are comprised of exons and introns. A gene does not have extragenic DNA within it. So I'm going to step away from the computer for a moment. So this right here, the left of exon A here, focusing on gene 1, and the right side of exon B, Like these are the bounds of gene 1 here. There's two exons here. There's an intron in between. It's not labeled, but that little black line is what qualifies an intron. There is no extragenic DNA within it. And the name extragenic kind of like implies that it's extra, outside of genic, of a gene, outside of a gene. So you would not find extragenic DNA within a gene, only exons and introns.
Yeah.
We're gonna define what those bounds are when we get to transcription next unit. Did you have a question? Cool. All right, let's keep moving. Oh, sorry, I didn't see your hand there. Last question. Go ahead. So say that one more time. This is percentage of our entire DNA. So extragenic DNA makes up about 3/4 of your entire DNA. Only 1% of your DNA actually encodes for the proteins that are in your body, and 24% of them are introns. Does that help clarify? Great. So if introns are within a gene, but don't actually encode for part of the protein that they encode for, what happens to those introns? What happens is this important process called splicing. So we have a gene here where there's exon 1 and exon 2 with an intron in between. During transcription, when we go from DNA to RNA, when we get that RNA molecule from when that gene that is transcribed, that RNA is sometimes called a transcript. You'll hear mRNA transcript, a transcript, but when transcription first happens, we usually refer to that first mRNA as the primary transcript. Those introns are there, but when we get to the final form of that transcript, when it's ready to contribute to translation and make the protein, those introns are not there in that final functional form of the transcript. So what happens here is we have the primary transcript up here to the top right. Splicing removes the introns, and what remains in the final functional transcript is just the exons in the RNA form. We are at the RNA level here. And so splicing removes introns to get the final functional RNA transcript that'll be used to create the protein that it encodes for. And so the big takeaway here is that splicing removes introns at the RNA level at this RNA transcript. Now, when we have, usually our genes don't just have two exons with like one little intron in between, but we have many exons within a given gene. Our genes are usually thousands of base pairs long. And so what ends up happening is that we have many exons with many introns in between them. What can happen is that there's different splicing events, the removal of introns, that can sometimes actually remove an exon and create different variations or variants of the RNA. And we call this alternative splicing. And so what ends up happening is that one exon for a given protein, bless you, can be removed. And when we get these different forms, these are called alternative splice variants. So we have here we have three exons here, A1, A2, and B. And we have these introns in between them, here between A1 and A2, and here between A2 and exon B. What ends up happening is that you can have different splicing events happen where when removing these introns, you can sometimes remove this exon and get this splice variant where it's just the A1 and B exons, or that's one version of this protein, or you can remove the A1 exon during splicing that just produces this other variant that leads to a different version of this protein. And so alternative splicing events can happen. The mechanisms that regulate this are not fully understood. There's still a lot of work trying to understand this, but what we do know is that it happens, that you can get different splicing events that can sometimes remove exons and produce different forms or variations of this protein. And again, these are called either transcript variants or splice variants. So, yes. Great question. It wouldn't lead to the formation of a totally different protein. It could lead to a dysfunctional version of this protein. It could lead to a less functional version or an overactive form of this protein. So sometimes different splice variants might not have a huge effect, but can have either a really harmful effect. Not either. I'll say it can have a harmful effect on the protein's function. Does that help clarify? Yeah, because if we think about the different proteins we've talked about in this class where there's different domains, it's perhaps possible that these domains are encoded, their function is imbued by a certain exon, and the removal of the exon might remove that domain. If we think back, for example, to the botulinum toxin, where it was one polypeptide with 3 domains, the loss of one of those domains, the alternative splicing, could totally render it non-functional, perhaps. And so it's good to be thinking about how different splice variants can potentially connect to your project, to some disease, or some biological process. So we have 3 flavors of RNA. There's one that I've already alluded to really briefly, that being messenger RNA or dubbed mRNA. This is the flavor of RNA that encodes for our proteins from a specific gene. And so the way that information is carried, and the reason why this is the case is, again, we'll talk about this a little bit more in the next unit, but our DNA is not just broken up into exons and introns and extragenic DNA, but within a given gene, every 3 base pairs is referred to as a codon. So to give you one example, we talked about in the innate immunology lecture that every protein begins, their first amino acid is methionine. The reason for that is because every gene has some start codon, these 3 base pairs. This is just an example. I don't expect you to know this now. I expect you to know that for unit 3. So I'm kind of exposing you to it a little bit early. But every gene has a start codon, A, U, G, or that's what encoded at the RNA level. And a helpful way to remember it is school always starts in August, AUG, August, start codon. But that AUG encodes for the methionine amino acid. So that's why every protein encodes for the first amino acid is methionine. And so three base pairs is a codon. Each codon encodes for 1 amino acid. And so that's what information is carried by mRNA. Another flavor of RNA is called transfer RNA or tRNA. This is a flavor of RNA that helps with the process of translation of producing proteins that is encoded in mRNA, and it actually helps to read the codons and bring over the amino acid to that corresponding codon. And so it helps decipher the code in our mRNA and bring the amino acid that corresponds to it. And finally, the last flavor is ribosomal RNA or rRNA. Ribosomal RNA, what it does is it helps to associate with certain proteins to form a ribosome that is the location where translation, the synthesis of proteins actually occurs. And so they all kind of contribute to the synthesis of proteins. mRNA encodes for it. Transfer RNA helps bring the amino acids over that corresponds to a certain codon. And ribosomal RNA contributes to the formation of a ribosome that is like the unit where translation occurs. And what that looks like here is we have mRNA down here at the bottom of the screen, those nucleic acids down there. We have transfer RNA that comes in, ribosomal RNA that helps with the formation of the ribosomal protein. The first transfer RNA comes in. This is the first one, so it's bringing in a methionine that's corresponding to the AUG right there that comes from the start codon in the gene. And so once that happens, you get the fully formation of a ribosome, and then you start getting more transfer RNA that brings in an amino acid. These yellow balls are the amino acid. They start stringing together to start beginning the formation of a polypeptide, and it just kind of shifts over and continues this in an iterative process. And so each of these flavors of RNA help contribute to the process of translation, the formation of proteins here. So if we were to go back to our slide on what the central dogma is, and revisit it and maybe provide a little bit of clarification here, we could say that DNA is transcribed into RNA. That RNA is spliced with the removal of introns usually. There might be some splice variants, alternative splicing, but in general, splicing removes the introns. That RNA that's been fully spliced is now translated into protein. That protein folds into its functional shape, and that protein usually undergoes some sort of modification usually. That can be the addition of sugars, that can be disulfide bonding, that can be some sort of phosphorylation event. But these, after translation, any modification, we call those post-translational modifications. And again, not really part of the central dogma, but a thing to consider is that DNA can also produce more DNA during replication as a part of mitosis. I'm going to stop here and give space for a couple of questions. Yes. Sorry, can you say that one more time? I just couldn't hear you all that well. Not that it's 3 codons, but that 3 nucleotides in RNA equals 1 codon. So if the reason why that's important is because when transfer RNA comes in to bring in that amino acid that it bears, it's reading 3 nucleotides at a time. So and again, we'll go into greater detail about this, but I can spend like 20 seconds trying to describe this in greater detail, but because we have a lot to cover today, I'm going to say that the take-home message for when it comes to codons is that it's a way how our nucleotides are read, just 3 base pairs at a time, 3 nucleotides at a time. So 3 nucleotides, it could be ATT, ACA, what have you. And in the case of the start codon, AUG, it's 3 base pairs equals a codon because each amino acid corresponds to a certain combination of codons. Does that help clarify? Yeah, go ahead. Sorry, say that one more time. Yeah, so that's a great question. So typically, mRNA comes from our DNA in the nucleus. There's also DNA outside of your nucleus, typically in your mitochondria. And so some subribosomal RNA, some ribosomal RNA is encoded in your nucleic DNA, but your transfer RNA, if I remember correctly, is encoded for in your mitochondrial DNA. Well, that DNA has a fairly different structure and different mechanisms. And so it's still transcribed. It still obeys the bigger laws here, but the way it's regulated is different than typical transcription. Does that help clarify? All right. So to apply, we're going to take some time to apply this to antibody diversity. So given what we know about genes, I asked you guys earlier about whether every different possible antibody could be separately encoded in a gene. The answer was no, because we have millions of possible antibodies, but only 20,000 genes. And so the way that happens is not by each gene antibody encoded for a gene, but a different regulatory process. And it's not also going to be via alternative splicing of our mRNA. That is a part of the puzzle, but that doesn't explain the entire diversity here. And so what I want to take a moment to hit pause on and explain to you guys is that what we're going to jump into now is, in my opinion, one of the more complex concepts in immunology. So what I want to recommend to you guys is, and what I'm going to try and do a good job of doing, is maintaining the big picture. So I've tried to make sure that I write out what are the different contributing factors to antibody diversity, and we're going to hit these 4 main concepts. But again, I'm going to try and do a good job of maintaining the big picture. And if I get lost in the weeds, please feel free to raise your hand and ask, what's the takeaway from this slide? If you ever feel like you don't know what question to ask, here's a couple of things you can ask. What's the take-home message from this slide? I don't understand this. It's okay to say that because it's not a mistake on your part if you don't understand something, but rather me not doing a good enough job of explaining something. So in the next 35 minutes, I'm going to try and explain what I find to be one of the more complex concepts in immunology, but I'm going to try and do it at a fairly high level so you can take home and understand the main concepts at play here, and hopefully trying to be able to see how this connects to the central dogma. So how your body actually accomplishes antibody diversity is empowered by a few important things. So the genes that encode for our immunoglobulins, or our antibodies, contain this library of possible DNA segments that are recombined together to form the heavy and light chains in our antibodies. So I'll take a step back and try and explain this a little more clearly. There are, during development, at the germline layer, when we say germline, that means in an egg and a sperm. When you hear somatic, you can think of that being, what's a good way to describe this? That soma means of the body. Somatic typically means things that happen to specific cells or tissues. When there's a germline mutation, that's going to be something that happens to every single cell in your body, because during development, every single cell originates from those early pluripotent stem cells when you're a blastocyst. So those germline mutations or changes happen to every single cell. However, somatic means typically it's restricted or specific to a certain part. Okay. You inherit a large possible library of different genes that can encode for the heavy and light chains. These are interchangeable. They can be swapped out during development when you're first creating your library of possible T cells and B cells. So the genome itself, when you hear the genome, that means the DNA. The DNA itself is actually rearranged to take these possible gene segments and move them around, piece them together to make a specific antigen receptor. And so this is happening during development. This is the antigen independent differentiation that we talked about last class. Taking a hematopoietic stem cell, it becomes a common lymphoid progenitor to a T cell or B cell. So during that differentiation, what happens is there's this rearrangement of your DNA. Another word for that is recombination. So whenever you hear recombination, think of like actual cutting and pasting together of your DNA. That's physically what's happening here. There's a cutting and pasting to take these possible DNA segments of these antibody or antigen receptor genes and piecing them together to make different possible combinations. Yes.
What?
Yeah, the splicing is happening at the RNA level, but recombination, I'm actually really happy you asked that. When you hear splicing, think RNA level. When you hear recombination, always think DNA level. That's gonna be a really helpful guide as we go through these remaining slides. And so the recombination is happening as a cutting and pasting that's happening at the DNA level. Splicing is a cutting and pasting at the RNA level. And so this is happening during development and helps with the formation of every possible T cell or B cell with their unique antigen receptor. So both when we think about antigen binding to our antibodies or binding to our antigen receptors, we have both the heavy and light chains involved in this process. So I have antibodies drawn at each side of the board here with the blue and red. I hope it's red. Forgive me if I got it wrong. Again, colorblind in case that might have been weird. Blue being the light chain, red being the heavy chain here. And they both consist of the FAB domain, this antigen binding pocket. They each have, and this I've tried to split it up here. The right side labels what we call their constant and variable domains. They each have a variable domain, and they each have a constant domain. As you can imagine, the variable domain is what is further up to where the antigen binding pocket actually is. And so they each have their own variable region or their own variable domain. What's important to note is, and again, this is kind of intuitive if you think about it, but in the actual antigen binding pocket right here is what we call the hypervariable domain, where the antigen, where it actually binds to the FAB domain, possesses what we call a hypervariable region. And so this is kind of intuitive but important to know. So what ends up happening is that, again, you at the germline level, so at the egg and sperm level, you inherit a library of different possible combinations of genes that can encode for your heavy and light chains for your antibodies. These different gene segments are are broken up into 3 parts for the heavy chain and 2 parts for the light chain. The 3 parts are V, D, and J, the variable segments, the diversity segments, and the junctional segments. This is for heavy chain. And so you inherit every single possible V, D, and J segment. So you get 300 possible variable segments, 30 possible diversity segments, and 6 junctional segments. During this, what we call VDJ recombination, there is a cutting and pasting of the actual DNA where you end up with one specific variable segment, one specific diversity segment, and one specific junctional segment. That, again, this is, you inherit all of these possible segments are inherited at the germline level, but the changes are happening in are happening differently in every specific B cell or T cell. And so that recombination is somatic. It's not happening in every single cell everywhere the same. However, it's happening differently in each one. So this is an example of somatic recombination. So in one given, for example, in one given B cell, there might be, the variable segment might be V1, D1, and J1. In the next B cell, it might be V2, D1, and J1. They're recombined differently from one B cell to the next. And because they're happening differently, because it's not ubiquitous in every single cell, it's somatic, okay? And so these different possible combinations is one contribution to the diversity of what kind of antibodies we can produce here. And this is specifically on the heavy chain. This is modular. There's cutting and pasting of different possible combinations here for the V segments, the D segments, and the J segments. And so what this recombination looks like is there's a cutting and pasting event where the segments that are going to be joined together are physically brought close to each other, and the remaining segments are then cut out to produce one specific V, D, and J combination that is on the heavy chain. I'm going to go here first, and then you ahead. We'll just go down the line.
How does the inheritance work exactly? That you can only have like, it says around 30 V, 30 V's, and 6 J's.
You said, how does inheritance work?
Yeah, like in terms of like inheriting the chain genes from like your parents.
Yeah, so great question. You're not inheriting, so you're not inheriting this specific gene segment. What you inherit is every possible variable segment, every possible diversity, and every possible function. Does that help clarify? So the germline inheritance is you're inheriting every possible one of these gene segments. What happens somatically in each individual B cell is this process where there is a cutting and pasting to produce in one specific B cell, one specific combination here. Does that help clarify? Okay, you want to go ahead? Yeah, great question. This is happening during development. This is happening as the baby is developing in utero. And so like this, what's a good way to say this? Yeah, there is a rule of thumb that at 2 years of age, there's like a fully formed immune system, for lack of a better phrase. but there's still this library of antigen receptors that are this library is developed enough to where before they turn 1, they're able to start getting vaccinations to start developing the antibodies to protect them against certain antigens. Does that help clarify? Do you want to go? Yeah, thank you for asking that. These are all genes. This is at again, so this is at the DNA level. They're each individual, they're each genes. I would say gene segments that contribute to the formation of the heavy chain gene. So, sorry.
And like, so you're just gonna go like a random B, a random B, and then a random J.
Yeah, there's actually an advantage to this being a bit random to the point where you want there to be every single possible combination that's developed. Did you have your hand up a moment ago?
Yeah. So then, the germline, they don't rearrange until they differentiate.
Yeah, so
They just
germline, you get a sperm and egg together to get that fertilized egg where it's where you get a blastocyst. You don't have a B cell or T cell quite yet. So they're not able to undergo BDJ recombination here because there are other proteins that help mediate this process. And if it's not a T cell or B cell, they don't express those proteins to help this to occur quite yet. Does that help clarify?
look like they just joined together from the sperm and egg cell, and afterwards in the semen, and the differentiate cell.
Yeah, you'll start getting with to make this as simple as possible, at the germline level, from the germ cells, sperm and eggs from your parents, you inherit this library. I'm going to toggle back. You inherit this library of possible gene segments that encode for your heavy chain or light chain. It's in your cells that not just from the germ cells from your parents, but in your cells, when you have T cells and B cells during development, that you will start having this VDJ recombination occur, where you get specific combinations that are unique to a specific T cell or B cell. I think I saw a hand over here. Yeah, go ahead. Is it totally random, like which V and which D and which J?
Like how many of each kind? Like, there'd be like 20 Bs and then one and 5 and then other.
It's just one. So maybe this will clarify. There's one V segment, one D segment, and one J segment. Does that help clarify?
Yeah, I was just wondering because like after the recombination, makes there's like multiple.
Yeah. So those will be removed so that there's one specific VDJ. The bringing together VDJ helps mark that this is the hyper variable part of the heavy chain that is at the antigen binding pocket. So, okay. Let's keep moving forward. So V, D, and J segments are present in heavy chain. Well, sorry, I'll take a moment to step back. What happens next? It's so inopportune whenever it happens. So these are the VD and J segments are part of the variable domain of heavy chain here. Further down, so if you look at heavy chain, the actual protein of the antibody, there's variable and there's constant domains underneath. Downstream of these VDJ segments are constant domains here that are encoded. And so you'll have, after transcription and splicing, you'll have your VDJ segments and your constant domains just downstream of that encode for your heavy chain of antibody. Light chain is pretty similar, just a little different. There's no diversity segments. And so there's 100 possible variable segments and 4 possible junctional segments. But again, this recombination is the same, whereby you have the bringing together of the V and the J segments here that help encode for the actual variable domain of light chain. So again, all of this is encoding what's right here, this hypervariable region. Again, there's also a constant domain. And so just downstream of that, there's going to be a constant domain for light chain. And it's also going to undergo transcription splicing where we have a V and a J segment and then a constant domain right underneath it. To try and illustrate this again, this is pulled from kind of like the immunology textbook, James Way Immunology or Immunobiology, forgive me. So both for heavy chain and light chain, there's all of these possible different V and D and J segments. For a given T cell or B cell, there's one specific V, one specific D, and one specific J that are brought together at the DNA level with the constant domains just downstream further down. It'll be transcribed. You'll have the introns spliced out. And then after translation, those segments then encode for either the light chain of an antibody or the heavy chain with their constant domains just afterwards. And so I thought this might be like another helpful illustration of what this all looks like and what this leads to, connecting the DNA level to the RNA level to the protein level. So when we think about the degree of diversity here, with the heavy chain, we have 300 different variable, 30 different diversity, and 6 different junctional. That leads to 54,000, approximately 54,000 different VDJ combinations for heavy chain. I should have changed that. For light chain, there's 400. You can tell last year I might have gotten a little lazy with just having students do the combinations. Yeah. I also was a little sassy when I wrote that last year. So for light chain, there's 400 different possible combinations, given that there's 100 different variable segments and 4 different junctional segments. When you multiply them together, it's approximately 20 million different combinations of the different heavy chain and light chain possibilities you can have. And so this is a great degree of diversity, but not quite yet what we need to be able to respond to every possible antigen. And so what ends up happening is that we need to provide more diversity here. And so I'll say, usually when you see VDJ recombination, there's parentheses because light chain doesn't have it, but we have VDJ recombination that we've talked about. The next level of diversity that contributes to our combinatorial possible diversity is called junctional diversity. What that means is that the joining of the V to the D and the D to the J, the way that happens is not quite precise. What ends up happening is that the sequences for each of them, when they're joined together, they can be joined together in a few different possible ways. There's kind of this random sampling of nucleotides when segments are brought together. And so if you take a moment to look at how these different V and J segments for a light chain, because it has light chain D segment, when they're brought together, there's a changing of what nucleotides are cut out or included. And each of them encode for, again, 3 nucleotides equals 1 codon. 1 codon encodes for an amino acid. Each of these codons, so CCT encodes for proline, but the changes here in the middle encode for different amino acids that is represented in that middle column for the amino acids there. And so the way these segments are brought together, they can be joined differently. That also provides another layer of diversity for what possible amino acids, therefore what possible antibodies can be generated. And so because this is happening between the V and the D and the J, and then the V and the J, you have 3 sites at which this can happen with seemingly different possible junctions that are formed here between these segments. And so this joining continues to happen until it's successful. And then this varies a little differently between light chain and heavy chain, but there's a little bit more complexity to that. And so what the take-home message that I want you to walk away from this slide with is that the way these segments are joined together is imprecise and not perfect and won't always be the same between a given V and a given between two given segments, and contributes to another level of diversity that we call junctional diversity. Yes.
It's not always four though, right? Like it could be like for a span of like 9 different.
It's not like four possibilities. Yes, yeah. So it's this is just like one. This is just like a list of four possible examples. So you're right. It's not always just going to be four. There are the number of possibilities is kind of what's a good way to say this? The number of possibilities is kind of restricted based on the size of the V and the D segments. But you're right. There's more than just four possibilities there. Yes. Yes. Yeah, that's going to contribute to the formation of a heavy chain or a light chain. Yeah. Yes. So yeah, both heavy, thank you for asking this. Both heavy chain and light chain have a variable domain and a constant domain. The main difference between heavy chain and light chain is that heavy chain has two more, sometimes three more, based on what class of antibody is, but it just has more constant chains. So if you see here, we have on the light chain, variable light, constant light. On the heavy chain, we have variable heavy, constant heavy 1, constant heavy 2, constant heavy 3. So this is what an IgG class of antibody would look like. If this was IgM, there would be a fourth constant domain. I think it's IgM, fourth constant domain. But in general, yes, this is what that's one of the main distinctions. I think I saw another hand a moment ago. All right, we'll press on. So we've talked about VDJ recombination. We've talked about junctional diversity, the different imprecise joining of these segments together. Another form of diversity here is mutations that can happen after an exposure to a cognate, a specific antigen. So when we have a B cell that binds to antigen, presents it to a given T cell, and if there's proper activation, and that's the appropriate epitope there, there's going to be cytokines that release, that activate these cells. What ends up happening is that this B cell differentiates into a plasma cell. That's the professional antibody producing differentiated B cell. And so there's going to be DNA replication that occurs during proliferation. And it is a massive amount of proliferation. Think about the last time you had the cold or the flu, and think about the lymph nodes that were really tender and swollen. That's going to be B cell and T cell massive proliferation, or I should say also maybe the last time you had strep throat for the higher lymph nodes there along the jawline. Those lymph nodes, when they're tender and swollen, that is T cells and B cells expanding, proliferating massively. And so because they're proliferating, there's going to be mitosis, there's going to be DNA replication that happens. During that DNA replication, there's going to be some small errors that happen amongst those different B cell clones that are specific to that one antigen. The V segment, the variable segments in particular, might have very specific little mutations that can happen during DNA replication. And again, because it's happening within not every single cell in your body, that would be a germline mutation, but because it's happening, A, after development, but B, happening among specific cells, it's somatic. So these are somatic hypermutations after antigen exposure during the expansion of a certain B cell clone, where these somatic hypermutations actually have a protective, helpful and evolutionary protective role, where perhaps these different mutations that happen can actually lead to a slight change in the B cell receptor or in the antibodies produced that actually bind a little bit better to their sibling progeny, if that makes sense. And so there's going to be competition amongst these different small errors to try and develop a more refined antibody or B cell receptor that better binds to that specific antigen. And so it's kind of survival of the fittest of these B cells that all originated from the same clone that recognize that specific antigen, but each of them might have like little small changes, little small hypermutations that perhaps one of them will outcompete the others and bind the best so that that specific clone can be preserved as a memory B cell and your antibodies from that B cell will endure. So to kind of see what this looks like, we have a B cell that is it's an antigen presenting cell. It bound to a specific antigen, displayed it to a T cell. There's cross activation. That specific B cell now will proliferate massively. What's going to happen is we perhaps might have one specific hypermutation in one of them. We'll call this B sub X, where that mutation might lead to a lower binding than the original B cell receptor or the antibodies from these B cells, or perhaps B sub X will bind better. And so when there is subsequent exposure to that antigen, there's going to be basically these different hypermutations will lead to a competition whereby perhaps B sub X binds the best and will bind first to that antigen, and then B sub X will present to T cell, expand, and then you'll have B sub X expand even further. And there might be, again, maybe a small somatic hypermutation that happens as a result of those B cells now expanding. And so this is a way that you can get another degree of diversity in your antibodies that is in response to an antigen, but these small hypermutations that happen somatically, they're not germline, they happen in these specific cells, so it's somatic, these specific small mutations. Whenever you hear hypermutation, think of like a very small change, a very small error that can actually lead to a better outcome, better performance of those B cells. I'm going to stop and give space to any questions there. Yes. That's specific to cytotoxic T cells, the CD8 positive T cells. B cells are an antigen presenting cell. So they express MHC class 2 that they display the antigen on. And so they're only going to be displaying that to a CD4 positive T cell, or a helper T cell is another name for it. And so when a B cell displays to a CD4 T cell, there's going to be signals sent back and forth to one another. Some of those signals are going to be signaling proteins. Some of them are going to be, and this is outside the scope of this class, but more like a graduate level immunology, surface proteins that are expressed that also serve as activation signals with one another. But the key thing to remember is cytokines, these like released signaling proteins that activate each other, because that's the main way in which these cells activate each other, that lead to B cells differentiating into a plasma cell, the professional antibody producing cell. They'll proliferate massively, and then they're going to stick around for a subsequent exposure. When there's a subsequent exposure, perhaps one of those B cells that proliferated had a small somatic hypermutation, a small little change to the variable segments on the heavy chain or light chain that could lead to a better performing B cell clone. And in this case, it did, so it binds better. And so now you get the antibodies from B sub X, this better performing B cell that lasts and endures. Okay, so another form of diversity is called class switching. So there are two ways. So we talked about the different classes of antibodies: IgG, IgM, IgA, IgD, and IgE. When it comes to their abundance, a quick trick to remember is GMADE. GMADE, or the IgG is the most abundant. IgM is the second most abundant. IgA, IgD, and then the last but not the least important is IgE. That's the one associated with your asthma and allergies. I spent a lot of my time in my PhD looking at IgE activation. So it's the least abundant, but it's like not the least important. I just want to say that really quickly. The decision between which of those classes of antibodies, the way in which they're determined is determined by two key ways. So in our adaptive immunology lecture, the first one that had to be recorded, there is small pieces of information about each class of antibodies. Two of them, IgM and IgD, are key to note really quickly because they're both the flavor of antibody that makes up our B cell receptor. But what's key to note is that they're also, IgM in particular, is the first class of antibody released during response to an antigen. And so what ends up happening is that for our heavy chain and light chain, we first have, and again, a lot of you have already taken semesters of calculus and physics, and so I'm sure everyone's Greek is strong. But these Greek letters in the constant domain help determine what class of antibody this is. C mu encodes for IgM, C delta encodes for IgD, and those are the first two constant domains in heavy chain. And again, based on someone asked this question earlier, the FC domain determines what class of antibody this is. Because the FC domain is only associated with heavy chain, it's the heavy chain constant domains that dictate and determine what class of antibody this is. And so because IgM and IgD are typically the most common, part of the reason is because C mu and C delta are the first two constant domains in the constant region of the heavy chain gene locus. So what ends up happening is that when we have a specific VDJ recombination event that occurs, we also have C mu and C delta here. What ends up happening is that there can be differences between whether it's IgM or IgD that is explained by alternative splicing alone. And so what that looks like is we can have some B cells that have IgM on their surface, or some B cells, their B cell receptor can be an IgD. Again, typically when antibodies are secreted and produced, it's first going to be as an IgM, so it's going to be typically C mu, that is a constant domain for secreted antibodies. But when it comes to the antibodies that make up the B cell receptor, some of them are IgM, some of them are IgD. That's dictated and determined by this alternative splicing that can happen. What's important to note is that the antigen specificity does not change. Does anyone want to venture a guess why antigen specificity does not change here? Yes. Exactly right. The splicing is occurring with the constant gene segments. VDJ, the variable domain, still the same. Alternative splicing does not affect it. Okay. This is alternative splicing happens at the RNA level. Recombination is at the DNA level. So this is splicing that's only changing the RNA, whether we see C mu or C delta as the constant domain here. Yes.
How does this lead to diversity? Like the variable domain is something that
if you think about the different possible antibodies that can occur for antibody diversity, this only adds like a small degree of diversity. The next slide will add a little bit more information about the different flavors of antibodies that can occur. But best case scenario, because we have 5 flavors of antibodies, the diversity is multiplied by 5, based on what we talk about in the next slide. So that was alternative splicing for determining whether IgM or IgD at the RNA level, what the constant domain is. However, there is also recombination. Again, when you hear recombination, think DNA. There's also recombination that occurs that also switches the classes of these antibodies. So after VDJ recombination, we have our VDJ variable segments here. We have all of these possible constant domains. Again, mu is IgM, delta is IgD. All of the gammas encode for IgG. IgA, that's mucosal, the mucosal class of antibody for IgA. Epsilon is IgE. And so when it comes to what class of antibody, we first have mu, or IgM, sorry, because mu is the first constant domain that you see listed there. But when you have a subsequent exposure, a second exposure to a given antigen, you're going to have a class switch that occurs. So again, IgG is the most abundant class of antibody you have in your body, because typically when you have subsequent exposures to a given antigen, they will switch from IgM to IgG typically. That's why it's the most abundant class of antibody. And so what ends up happening is that there's recombination where now the mu segment will get cut out, and we have these different constant domains that are possible here. And the first one there is C gamma 1. And because of that, IgG1 of all of the IgGs is the most abundant antibody in your bloodstream. And so that will typically lead to this class switch where your antibodies will go from IgM after first exposure to IgG1 upon second exposure. Big picture take home. I'm going to take a step back. There is the previous slide was alternative splicing between IgM and IgD. It's all splicing. It happens at the RNA level. This is class switch recombination. So this is DNA level changes, recombination, that can dictate and determine what class of antibodies you will get from a given variable domain. And this also explains why IgG1 in particular is our most abundant antibody in our body. And so this is and so once we have that, the association of our heavy chain and light chain, the different combinations, this is the final degree of diversity. The different combinations of possible heavy chains and light chains, the way in which they come together can vary differently from one B cell to the next. And so adds the final layer of diversity that helps us determine how many different possible antibodies that we can produce. And so all of this is there's another question. Don't worry about that. All of this helps to explain how we can achieve antibody diversity. Defects in this particular in VDJ recombination lead to some instances of severe combined immunodeficiency, or SCID. One is T cell and B cell SCID. This is specifically targeting some of the proteins that facilitate this recombination. Another example is Omen syndrome, where again, it's another defect in recombination chaperone proteins. This can also actually lead to some instances of cancer, specifically lymphomas. Lymphomas are mutations and cancers that occur within our lymphatics, particularly lymph nodes. Burkitt's lymphoma takes a oncogene. Whenever you hear oncogene, think of a gene associated with functions of tumor cells or enable tumor cells. And so MYC drives proliferation. Tumor cells love to grow and proliferate. This is a mutation where MYC is tied in with one of our immunoglobulin genes that leads to Burkitt's lymphoma. Follicular lymphoma or B-cell leukemia. Leukemia is typically a cancer of specific immune cells. This is where BCL2, a apoptosis regulator, is translocated into one of our junctional segments for our antibodies. If antibody diversity really stuck with you, or VDJ recombination, we have a great group of people here at UVA who are studying this space. I apologize for going a couple minutes over, but I'm going to stay back for any questions. Thank you for your time.